All right, today we're going to talk about one of the coolest, most elegant tricks in the entire genetic engineering playbook. It's called blue-white screening, and it's this brilliant way for scientists to literally see the results of their invisible work, basically turning a super complex problem into a simple choice between two colors. So picture this. You're a molecular biologist, you're in the lab, and you've just run this big experiment to stick a tiny little gene into a piece of bacterial DNA. The problem? Your chances of success are ridiculously low. So how on earth are you supposed to find that one single successful clone in a massive sea of failures? Well, the solution is it's as simple as it is genius. Instead of trying to find that one tiny needle in a giant haystack, what if you could just paint the entire haystack blue? All of a sudden, that needle, your successful experiment, just pops. It stands right out. And that one simple visual cue, it changed everything. To really appreciate just how clever this trick is, we first have to understand the sheer scale of the problem we're dealing with. We are literally hunting for a single successful event happening at the molecular level. Okay, so the basic goal of gene cloning is pretty straightforward on paper. First, you take a little circular piece of DNA from a bacterium, that's called a plasmid, and you use what are basically molecular scissors to cut it open. Then, you get your gene ready and you use a kind of molecular glue to paste it into that open plasmid. And finally, you get this new souped-up plasmid back into bacteria so they can make millions of copies of it. But here's the catch, and it's a big one. After you cut that circular plasmid, the two ends you just created are sitting right next to each other. And they are way, way more likely to just find each other and glue themselves shut again, leaving your precious gene of interest totally out of the loop. It's a super frustrating event called recircularization. And this slide, wow. It really lays out the tough numbers we're up against. In a typical reaction, you'd be lucky if maybe 5% of your plasmids actually take in the new gene. The other 95%, that's just the original empty plasmids that snap shut. So when you grow all this on a petri dish, you're looking at a situation where 19 out of every 20 bacterial colonies are failures. So this is where the color coding comes to the rescue. Faced with this incredibly low success rate, scientists developed this beautiful biological hack to make the needle in the haystack just jump right out at you. The big idea, the real light bulb moment, was this. What if we put the spot where we insert our gene right in the middle of another, non-essential gene? We can call this a reporter gene. So if our gene insertion actually works, it physically breaks or inactivates this reporter gene. And here's the magic. This reporter gene has one job and one job only, to produce an enzyme that creates a bright blue color. So think about it. If the plasmid just closes back up on itself, which is a failure, the reporter gene is totally fine, it works, and the bacterial colony turns blue. But if your gene of interest successfully gets in there, it breaks the reporter, no blue color can be made, and the colony just stays its normal white color. All of a sudden, failure is blue and success is white. So how does this actually work? What's going on at the molecular level to make this blue or white decision? Well, let's meet the key players in this little biological drama. First up is our enzyme beta-galactosidase. It's encoded by a gene called LOXC. Now, its day job in E. coli is to break down lactose, milk sugar, for food. But for our purposes, the really important thing it can do is cut up a special man-made chemical. And that chemical is called xgal. On its own, xgal is totally colorless and visible. But when our enzyme, beta-galactosidase, gets a hold of it and chops it in half, boom, it turns into this really vibrant blue compound that won't dissolve. This is our ink. This is what's going to paint all our failures blue. Now, the real secret sauce, the core mechanism that lets us control all this, is a truly clever bit of science called alpha complementation. It all starts with a special strain of E. coli bacteria that has a deliberately broken, non-functional version of that beta-galactosidase enzyme. But, and this is the key part, this broken enzyme can be rescued if you just give it one tiny missing piece, a small protein fragment called the alpha peptide. The best way to think about it is like a two-part security key. The bacteria has the main part of the key, we can call that the lock, but it's missing the teeth. The special plasmid we're using is engineered to provide those missing teeth. That's the fixer alpha peptide. When those two parts come together inside the cell, boom, you have a fully functional key, a working enzyme that can turn on the blue color. So here's the most crucial point of the whole system. Scientists engineer the plasmid so that the spot where you insert your gene, the cloning site, is located smack dab in the middle of the gene that makes that little key piece. A successful insertion literally snaps the key in half. It can't fix the lock anymore. Okay, so let's see how this all comes together on the Petri dish. This is that moment of truth, where all that clever molecular machinery pays off with a simple, beautiful visual result. This table just sums it all up perfectly. Let's look at the failure scenario. No gene gets inserted. The plasmid's key piece is intact, it hooks up with the bacteria's lock piece, you get a working enzyme, and the colony turns blue. Now the success scenario. Your gene is inserted. It breaks the key piece, no functional enzyme is formed, and the colony stays white. Blue means bad, white means good. The practical result of all this is just a beautiful, clear signal. The scientists can just look at the plate, completely ignore all the blue colonies, and just pick the white ones, knowing that those are the ones that almost certainly have the new gene they worked so hard to create. Now, as powerful and amazing as this technique is, it's really important to remember that in science, nothing is ever 100% foolproof. Blue-white screening is a fantastic tool, but it's got some important limitations you need to know about. First off, let's just be precise for a second. Blue-white screening is what's called a screen, not a selection. And there's a big difference. A selection would actually kill off all the unwanted blue colonies. A screen, on the other hand, just lets you visually tell the difference and pick the right ones from the crowd. The failures are still there, they're just colored blue. And there are a few ways it can go wrong. Sometimes, a random mutation in the plasmid can also break that key piece, which gives you a white colony that doesn't actually have your insert. That's a false positive. And sometimes, though it's rare, a really small insert might not fully break the key, so you get a blue colony that's actually a success, a false negative. And you can also get these tiny little satellite colonies that are just kind of piggybacking nearby. Because of all these potential issues, blue-white screening is almost always used as a first pass, a first look. As this quote says, you always have to follow up and verify your white colonies. Scientists will pick a few, grow them up, and then use much more definitive methods like PCR or even DNA sequencing to confirm they've got exactly what they think they have. Make no mistake, this technique was an absolute game changer. It took a really tedious, difficult problem and turned it into a simple visual task. It sped up research and enabled all kinds of breakthroughs, from making human insulin in bacteria to developing new crops. It's a biological reporting system that turns a molecular event into a color you can see with your own eyes. And that leaves us with a really fascinating thought. What other complex, invisible problems could we solve if we could just find the right way to color code the answer?